It's my pleasure to introduce Marco Molina. Marco comes to us from Brazil. He's been here actually a number of years at Texas A&M, working, as many of you know, in the multi-crop transformation facility. So you may have heard fellow students give their thanks to Marco for all the efforts he's helped with transformation across a number of different species. And so today he's going to focus on his rice work. So Marco.
Hello, good afternoon, everybody. One more time, thank you for the opportunity, and I will try to show a little bit of the work that we do in my project and also in the lab that I'm into and supporting Dr. Thomson and everybody else that actually helps a lot. So just some examples of some of the crops that we are working to. Today I'm gonna talk a little bit more on the basis of why step by step we plant transformation tissue culture. It's like one word, tissue culture dash transformation. We can see everywhere. This is how it works. And specifically, we're going to talk a little bit with our project in rice with the use of CRISPR-Cas9 approach. And then later I'm going to talk a little bit on the hemp project as well, like initial steps, but something with hemp as well. Few nice pictures with the process itself from the initial plant, right? Some halide formation, some somatic embryogenic formation, some GUS GFP expression. In here, our main goal is to regenerate a new plant from a tissue, from an organ, depending according to the species, with the variety. So this is our main goal. Always looking for this one, for the plant in the end of it. This is what we are looking for. And we just get into the biotechnology complex, right? So we are in the agricultural biotechnology, we have the industrial biotechnology, medical biotechnology, and in our efforts here, biotechnology plays an important role in that, and specifically after 25 years with the genetic modified plants, how they were very the impact that they have in the society from the producer to the consumer. So we have 20 years, 30 years of experience. They're there, they will come to stay. We are just going to branch out now. So maybe we are on the time to get a new breakthrough in biotechnology in our field. So we have to be ready for that. So the main species here that we consider, that we have spread out around the world. And thinking about biotechnology, it's tissue culture and transformation, tissue culture dash transformation. It's one of the tools that it requires. It's one of the tools that mostly required to implement that goal, to implement that biotechnology branch in the agriculture field. So, and it's very important. And as you guys can see, those are some examples of dedicated transformation facilities spread in some of the educational institutions here in the US. It's a little bit not up to date, but it's just the dedicated facilities that work with that provide that kind of service, that provide that kind of research and dedication that's specific in how to put those plants through the process. So as you also know, guys, that the big ones, the big companies, they also have a large research and development departments, a lot of people working on that, high level, a lot of money, a lot of input. So everybody's working in that sector. And also the startup, it's very recent. We are having a lot of requests from those small companies. They need our service. They need our help. When I say ours, from the academia to help them to get to that biotechnology, help them to produce a new or novel product, a new crop, that new plant. They require our help in doing that. And just in thinking in tissue culture, it's very amazing how it took almost 60 years from the first initial idea and say, hey, I think we believe, I understand, based on my research, I think that we will be able to produce a new plant from an organ, from a tissue, from something else. And that took almost 60 years to get to that. And then they proved the concept in tobacco, where they were able to actually regenerate plants. And from now on, from that moment on, the explosion of research, the explosion in how to do those things, the varieties that they were able to do, the species, and year by year in the past 4 decades, we have a lot of improvement in every single stage of that process, which is really cool. And we are just part of that. And this is kind of some of the team of the thing that I'm talking is like a one step at a time because it just takes forever. One example of the cycle, one example of the process, it's here in sugarcane, what we start with, and just to read out, it's the aseptic culture cell tissue organs and components under the defined physical and chemical conditions in which. So it's something very clean, super sterile, in a specific physical container with specific chemical composition during the full process. So we can start from the greenhouse. We can start from the field. In this case, we are working with ethical meristem. Ethical meristem goes. You need to clean up this material first, and then go through the process of growing collide. There will be that mass of cells, different non-specific cells. So there will not be ethical meristem anymore. There will be like a undifferentiated cells. So all that process. And again, our project here, our goal here is to get that beautiful regenerate plant, isolate those plants, and hopefully later on check with molecular analysis that plant will be a transgenic. If we introduce the transgenesis here during this process, right? And some of you examples, most of you guys are used to with Arabidopsis, fluoroclip, take up to 3 months to do it. We have a bunch of examples here that are around that, 3 to 4 or 5 months. There's something that is more straightforward from that process. And in the other end, we also have things that are much more complicated. Not so complicated, just take long by the nature of the species. When we're talking coffee, just this tissue culture process took us 24 months to do it without even trying transgenesis. Right, so that's another level of difficulty that they might have. And yeah, this is just an example of sugarcane and the steps of the process. We will show a little bit more later. And again, we have a lot of challenges and a lot of benefits to use tissue culture gas transformation. On the challenges, we know it's crop specific, it's variety specific. Often takes a long time whenever you consider those 6 up to a year to achieve the regenerated plant. It's intense work along the process during the subculture. Often low efficient, and often the available literature requires some experienced technicians to do that thing. It's not everybody that likes to spend a full day under the laminar flow. If there's just a specific person that can do that, like Marcella and Myra, they do all that kind of work. It's not everybody that likes to do that. But we also have benefits into it. Some of the challenges along the process, you're waiting 8 months, and maybe you can ruin your experiment with some contamination. The things go bad, the plants go down. So we try to minimize this as much as we can. Eventually, we have some contamination, but we can manage. We just have to be aware of it. So among the benefits, we can do a large scale propagation, micropropagation. That can be constant around the seasons. We can maintain indefinitely pretty much an important germinal plant that we need to save for some reason. We can generate disease-free plantlets. And the most important one here in my point of view is enabling the transformation, enabling the gene editing, enabling to think with, to work with the work either for the basic research or to generate a commercial product with the transformation. So that technology enables that. Right? And an overall vision of what happened, of how we can use is from this plant, we can use several organs, tissue, parts of those plants. So here on the top, we can use with the apical meristem, very responsive material to work with. We can use leaves, cotyledons, roots, and that allowed us to go to several pathways. Either we can also the internodes. We can either go to a direct morphogenesis, like directly from that tissue, directly from that organ, we can regenerate a new shoot, a new plant. or a somatic embryo. A somatic embryo is just like an embryo that is not coming from the sexual cell. It's just like the seed, but it's not the seed. It doesn't come from that cross. It just comes from the somatic cell. It's any other cell that is not sexual cell. Also, plants, some species, right, they prefer a different way. Sometimes they go through the direct morphogenesis, is what it just said, or indirect morphogenesis, when we have to generate that cowhide first, the cows first, that undifferentiated mass of cells that we can also go through a direct regeneration process or have to pass through the embryogenic process as well. And again, our main goal is to get that plantus, to get that seedling in the end. During the entire process, along those pathways, in several, where we see the stars, we can add the transformation. Then we can add the gene editing. It's there our point of interest, where we're going to be able to transform that cell or that tissue or that organ. So, and this is how things get tricky, because plants, species, varieties respond differently, and that will directly impact your ability to regenerate our goal, that liver plant. And then when we add the transformation, simplistic way of see the transformation is pretty much get that organ, get the tissue, put in contact with the agrobacteria in this case, do the subculture through a selectable media, to selection media. In that selection media, we can select which one of those plants will be our transgenic event, a putative transgenic event that will have the insertion on their genome. And again, that's how we are focusing. We're focusing on get those plants. But to get those plants is tricky. Sometimes we count with non-model plants, something that is more difficult to regenerate. Sometimes we don't have that plant at the right timing. Let's say, for example, we need at the right timing. In rice, for example, we use immature embryo. What is immature embryo? It's just a seed that is not mature yet. So we need those seeds. They're probably going to have on the right stage, 2 weeks of the year of the season. During 2 weeks, are you going to plant all your full experiments in those 2 weeks? Are you going to be able to get the amount of material? What happens if your experiment goes bad in the next 2 years? Then you have to wait a new season to get that. So that's a problem that can happen in that process. The methodology as well, and the transformation, selectable markers plays a role. And again, when you talk about CRISPR efficiency, also that efficiency decrease, and you have to have a very well implemented protocol to overcome all those problems. In more detail, or in more just explanatory way, explain the delivery, how the regeneration works, and our end goal in plan. As an example of the beginning of the process, plant and the species, the plant, the crop, versus the plant that we use. You guys can see the variety of the species. On the side, you can see the variety of plants that they are used. Sometimes we use cotyledon, sometimes we use the embryonic cowhide, sometimes the meristem, and so on. Those differences are based on the age of the plant, as in the case of rice. Sometimes the plant size. So what kind of interaction a little piece of leaf will have with the media versus a larger piece of meat would have interaction with the meat. Will be dry out faster, will be the exchange will be faster, the exposure to the hormones will be different. Yes, they are. And they're really, really playing an important role in why you have those differences. The orientation, the excised position, we put adaxial or abaxial, believe. Does it make a difference? Yes, it does. So in this case, there are a lot of variations. Just on the transgenic delivery itself, as we know, those are the main well-known agrobacterial mediated transformation and the biolistic one. Also, they play an important role in your results. And how, for example, agrobacterium, the strain that you're using is very important. It's more virulent, it's less virulent, colonize better the cell, the plant cell, or less. The concentration, the size of your construct. the infection is working or not. But at least it's the same. What size of your particles and it's the pressure, how distant are you from the tissue? Everything plays an important role. Regeneration itself. Remember when we talk about the pathway that go? So the pathways also can give you different responses. Either you go to a direct regeneration from the tissue, or you have to go to the calli formation first. And as you expected, all the hormones plays an important role here on this formation. Are you going to go through the biogenic, somatic biogenic pathway? Also, it's important things to consider. And talking about the formation of calli, the hormone specific, the most common one, auxins and cytokines, they also play an important role. If you put, for example, at the beginning, a lot of auxin, less cytosine, you're going to have more root formation. The opposite, less auxins, more cytosine, you're going to induce more shoot formation. What is the right concentration ratio along the process? Have to figure it out. And as you expect, it will respond differently according to the species, variety, and all the conditions. Factors also, more important factors, media composition, macro and micronutrients, the agent, the solidified agent. There are some tissues that just don't like too much moisture, too much liquid. You have to be better, you have to dry out that environment. Temperature, humidity, light, and the antibiotic and the selectable marker, and the selectable marker, like antibiotic or herbicide, also will affect your tissue. What we are trying to do here is to establish, can we establish novel tissue protocol for new varieties? Can we do that? Can we establish new protocol transformation protocols for that? That's what we're going to try to figure it out. We have a problem, we have the variabilities, we have the literature, we have how to design experiments. We have our hypotheses. We're going to test those hypotheses. We're going to measure those hypotheses. We're going to check if they are statistically different. We're going to reproduce all of them. If that happens, we have maybe a novel protocol, a novel something to contribute to scientific society. That's the idea. Some of the projects that I'm involved with, it's the first one, the rice one. We all know the importance of that food importance in the world and locally. For us here, this variety, Texas variety, the Presidio one that we are working with, all of it was based on a novel development of a novel protocol for that, using the gene editing approach. Second one, it's in hemp. It's an important agricultural potential. We are dealing with around 8 varieties so far, with novel scientific opportunities. Why? Because we have limited information. So challenging the unknown. That's the beauty. So that little information, when I mean little information, when you compare corn, soybean, other things that you have 50 years of research, 50 years developing in either in companies or on the university, right? Other way to see this is to get something easier to transform, like a tomato, tobacco, or something more routine basis, and try harder proof of concept on that. That's also on the map. And the last thing that I'm working on right now as well is on sugar beet. It's a totally new novel protocol development strategy. The information is very limited for that, but we're getting good results on that as well. With rice, it's a lot of the information. Rice is well known. It's a monocot. It's well known for transformation, but using the temperate Japonica varieties. So everything that we have in the literature is more for the temperate Japonica, which when we try to apply the already established protocols, didn't work. So that was not published, but was not easy to do. Supposed to be easy, but it wasn't. So we have to go for it because rice, because the Presidio is not like a novel, it's not a novel variety, but it still has plays a very important role in the production. And so we're able to optimize that protocol using mature seeds in that Presidio cultivar by using the gene editing validation tool. So we transform those varieties using the genetic technology, trying to achieve knockouts of those two genes, either the PDS or the YSA genes, which if we were able to knock out our phenotype would be like albino plant, albino regenerated plant. Why? Because those genes are important on the chlorophyll pathway, and so things just turn white. Just to show here that we will have to be checked. We're just checking if our guide RNAs were good to go, if our tools were validated. That's an in vitro test with ribonucleic proteins, where we have here 2 guide RNAs generated, and those guide RNAs, 2 guide RNAs for each approach, for PDS and for YSA. We have our template. So without the guide RNA, you don't cut anything. The guide RNA in complex with the Cas9, the available Cas9, you cut your template. So that means that they are going to the right place, they're cutting in the right place. That's what we expect to happen in vitro, in vivo. So we are just checking for both were positive. Again, for the YSA, our template, for the guide RNA1, our template for the guide RNA2, not cut and cut. We were successful of getting regenerated plants in both methodologies. We try also both methodologies, either agrobacterial and biolistic. We just choose agrobacterial to go forward with the further analysis and molecular analysis. That's we have around 60% in both PDS and YSA of regeneration of the plant. And overall, we just consider 12% with all the albino phenotypes, but when that number just increased because the number of initial explants was counted, but not the YSA was not counted as easy as the PDS. As the name suggests, I will show later, it's just that it's young albino. So the plant has to be really small. The 1, 2, 3 leaf stage, that's when you start to see the white characteristic. After that, the plant just recover and get fully green again. So that was tricky to analyze that. Just to show by sequencing that the majority of the mutations that happened were here on this side with the lesions less than 10 base pairs, either for PDS and in YSA as well. So less than 10 base pairs was the majority of the mutants that we got. And thus we have all the combination. We have alleles with the same mutation, we have alleles with different mutation, we have alleles with the wild type, and so we were able to regenerate a lot of things. That's just one example of, hopefully we can see, on the YSA, it's very tricky to detect the albino characteristic when it's very young. And again, after a week, you cannot see anymore. So, but we were able to detect all the variation by molecular analysis. Comparing to the PDS was very straightforward. You actually have your wild type, you have some variegation, you have phenotypes where you were able to regenerate full albino plants, but with problems in development, and you have like a normal plant, but full of albino. Okay, and this is just the process as a whole. There are a lot of work on this. The first one here is just we are talking about seeds, which is not, we don't have that information for this specific variety. So from the seed, we just go to generate produce calli. Those calli are different from the status quo of the project of the available protocols. Took a lot more time, even double of time to cultivation, took double time. And here the change in the hormone was also even up to 6 times more hormones in a specific phase to be able to regenerate plants, normal plants. So, but then again, a lot of tests, treatments, evaluation, and see which one would be the best response. In this case, works well with those main modifications. And also light and dark played an important role during the process. Every literature was saying, yeah, doing light, and then we try different approaches, and in the end, more dark period worked better for this variety. Okay, getting there. Remember that pathways that we were discussing? I just show how they work. This is what we are doing. This is the approach that we are having on the next project, that is hemp. We are trying to generate a new THC hemp line to make sure that in the field they will not be destroyed when they go over the threshold of 0.3 of THC. And we are we are supporting for startups. They want to achieve that commercial phenotype. And this is the approach that we are getting. We are trying several ways how to regenerate those novel plants. There are 8 new varieties we are working with right now. Main ones, cotyledon, hypocotyl, and leaf. So you expect that from every single initial plant, go through the calai production and then eventually regenerate one of the pathways. And they are working very well. You can see cotyledon here on the left, right? Here we go to the hypocotyl and then the leaf production. So we are still evaluating all of this. We are measuring which one will be the best approach. Everything is working well so far. The next step is just going through the transformation, right? The first thing we do is like establish a very good tissue culture process. Once the tissue culture is done, then we go to transformation or gene editing, whatever is the next step. And it's working. We have some standing dust here in 2 of the varieties. So the infection is working well. This regeneration, the regeneration is also working. We can see some spots with dust here. And the process is very similar. This is, I guess, for cotyledon. And this is how we see. This is an important step in the process when plants are already growing in little jars. This is the most important. This is where we are going to the molecular analysis. Once we can see roots growing in the selectome media, this is time to take a piece here and take to molecular analysis. This is where we are right now, where we can see our negative control, positive control, like plasmid control. We have some hits here for this variety. For the other variety, much better. We can see the positive control here, negative control, and samples 17 to 30 with some unknown, but the vast majority is just as expected. So we are having several good results on this. And right now, those plants are going through the greenhouse, room chambers, where they're gonna be evaluated in the phenotype if we're gonna actually trying to achieve those THC levels. So it's a next stage on this phase. We are hoping to see that. And with that, thank you very much to everybody, to Thomson, the lab, all the students that work, that help along with that, especially to Transformation Facility Mara and Marcela. They are doing a lot. And also all the support that we have for Live, SBA, and the other 2 startups that we're working. Thank you very much.
The mic's on now.
Give it a try. Did you have a question?
I have a question. The hemp work you showed the 3 methods. Is that with 1 company or 2 companies or?
That's the way that we try to establish a very good tissue culture process first. And then the company strategy we're going to use on the best results.
Okay.
So you mentioned there's different times, varying lengths of how long it takes to transform the plants. Is there any correlation between the difficulty of successfully transforming and the length it takes to do that whole process? Or is it just kind of random throughout?
No, I don't think it's a correlation of success. That's what it takes. It's what they require. And sometimes it's whatever. It's the process that either it's on the beginning of the process or to how to produce the right callus. Sometimes you have to spend more time in that phase, and then, or sometimes you have to spend a lot of time in producing the embryogenic embryo. For example, because of cotton, this is what we spend a lot of time. You have good calai, you have good everything, and you have to. The system just required to spend much more time to produce that embryogenic calai. So they need a lot of specific stimulants. specific conditions for that to happen. So you just have to spend time. It's not necessarily directly correlated with success or failure.
You had there was a range between, like, I think Arabidopsis was 3 months and coffee was 24 months.
Right.
And so do you have any idea like why there's so much of a difference? And if so, you didn't have hemp on there, so I was wondering how long does it take for the hemp?
Hemp is hemp is kind of in the same range, same emission rate up to 4 months, 3 to 4 months, yes. And this is part in the literature. It's just part of the recalcitrant nature of the variety of the species, which some require more conditions, either at the life formation or at the regeneration process. Just vary according with the crop, with the species that you're working mostly. Yes.
Is there one on the?
Yes.
How much do you guys charge per event?
You see, we work with several species, right? And that's totally related with how long they take. So sometimes 3 months. If you talk to me, sometimes 3 months. Hey, Mark, I want to work with something that is ready to go, easy to transform. Let's say about $1,000 to $2,000. Marco, it's something that I never tried. I have no idea how that's gonna respond. Nobody never tried. There will be a protocol development project that we're gonna work for hours, and the hour rates differently according to the difficulty of that process.
Nothing's for free.
We do things for free, for sure. We do a lot of pilot projects. Pilot projects like, hey, I have something here that I never worked with. Do you think this might work in something easy to do? Yeah, let me check can be in tobacco, can be in tomato. If the PI say, yeah, let's try to see if I have some expression, we do for free. Little bit.
Other questions? Any virtual questions? Well, let's thank our speakers again.